Hello, everybody. My name is Kathleen O'Hagan. I'm a facilitator within the ATMP forum here in BioPhorum, and you're all very welcome to this webinar that showcases a recent publication from the ATMP forum within BioPhorum entitled Material Readiness Guide and Assessment Tool for ATMPs. So I'll be the host for today's event, but I will be passing over to three of the team, three subject matter experts who were very much involved in this work, and we'll talk in more detail around the publication that we're presenting on today. Okay, so in terms of the agenda today, we'll give a little bit of background to the team that produced this deliverable, who they are and why they formed. And we'll also then share some of the key contents of our publication. So we'll start with the bill of material pre-screen. So this is an Excel tool that you are very much free to download, and we'll share that link in due course in this presentation. And that will help you quickly assess and prioritize materials within your bill of material. Also associated with that tool, the team have created a number of flowcharts and decision trees that will help guide you through some of the key activities for PPQ and commercial readiness. Again, you'll be able to download those, and we'll talk you through during the call today. Also, as part of this publication, we have made suggestions for phase-appropriate material specification requirements and specification templates. And again, we'll share some of our thoughts on that. Very quickly, I just want to take a minute or two to talk about BioPhorum and who we are. So BioPhorum is a global collaboration platform whose mission is to create a sort of safe environment where global pharmaceutical and device industry members can come together, connect, collaborate, and accelerate, and ultimately deliver change across the industry. We have over 150 member companies currently, and these 150 member companies consist of about 7,500 SMEs that are actively involved within the collaboration across 200 plus programs of change. In the last year alone, we've had over 170 deliverables, which include publications, presentations, and other resources that members of the collaboration can tap into. And in terms of, I suppose, what makes BioPhorum unique is we're very much member-led. We create a safe and confidential environment where our members can come together and allows for unfiltered discussion around challenges and possible solutions, and ultimately leads to cheaper, faster, and better journeys for all. So on today's webinar, you're going to hear from one of the teams that are part of the collaboration, and you'll see some of the contributors on this screen now. So we've had 15 member representatives across 11 organizations. I know a few of these are on the call today, so I just want to take a moment to say thank you so much for your great collaboration, and it really has been a great team to work with. And we do have three of those authors and collaborators on the call today who are going to talk you through the publication that the team have produced. So I think you all have your cameras on, don't you guys? I'm going to pass over to you guys. Maybe you can give a quick hello and introduction, and then we'll get into the presentation.
So my name is Monica Simons. I work over at Fujifilm Biotechnologies in Texas, and I'm in process sciences right now, and I specialize in raw material specifications.
Chandel Han. I am in Vertex Pharmaceuticals. Recently moved from Boston to London, and I am in the MSAT team, and a significant piece of my work is supporting raw material work.
I'm David Evans. I'm with Astellas Gene Therapies, and I work in raw material chemistries.
And you're all very welcome. So thank you so much for presenting today. Okay, so I'm going to pass over. We're going to sort of tag team, I think, with this presentation. I'm going to pass over to Monica to introduce why the team was formed.
For sure. So background to BioPhorum ATMPs, Material Readiness team. Basically, when you're dealing with product development, you're using a lot of raw materials. It can be media, cells, containers, tubing, all sorts of reagents, any number of things to create your medicinal product. And the thing of it is, you need to make sure that all those materials are appropriate safety quality to ensure that your product is also safe, high quality, and imparts the desired effect that you want it to have. Now, one of the big challenges of ATMPs is that a lot of times the materials that we use are not well characterized. They may be very unique. They may be in short supply. Any number of things could be challenging with the kinds of materials that we use. And so when you're trying to prep your material to go from your clinical to your commercial phases, there's a lot of factors that need to be taken into account with each and every material you're using in your process. And so because it's very complex and because there's a lot of different factors, you need to really consider which materials you need to work on, figure out timelines, figure out exactly what activities you need to do. And all of that can be very complicated. It can be very it's like, just like, where do you start? It can be very overwhelming. So when you have this whole list of materials and you're not sure where to start to make sure that your materials are basically up to snuff for your regulatory agency, you can run into issues like getting warning letters. Maybe you can't get your product to market on time and somebody else beats you to market. Things can get delayed. You can have challenges in your supply chain. A lot of different issues can come up. And if you don't plan appropriately, then your product can kind of not be doomed to fail necessarily, but can just kind of run into a lot of issues that will cost a lot of time and money ultimately. So why are we in this predicament? Well, it's because the medicines that we work on are very cutting edge. They're at the at the very boundaries of knowledge and technology, and we're constantly making new leaps every day. And so because we're making all these leaps and bounds in our knowledge and capabilities, there's that lag between what we need from our materials. There's a lag in experience because we don't necessarily, if a particular modality has only existed for past 5 years, well, whereas other modalities have existed for 10 or 20, you're going to lack that experience to, know how to really handle these materials. Also, regulations tend to lag too. So, as we make these leaps and bounds, the regulations are going to take time to catch up. So it's really hard to discern, like, yeah, you want to get your product out and help your patients, but it's so new that we don't really know what the rules need to be in order to have appropriate safeguards. But also it's all hard because a lot of these therapies take a lot of money and investment, and you don't want to waste that time and money, but you also need that capital to be injected in to like move forward. So it's just really hard to keep up with all the shifting and advancing factors. So what can we do about it? Well, we can kind of work with what we know, and based on that, we developed a tool to kind of help give a high-level overview of your materials. And in this case, I'll refer to as a BOM. So BOM refers to bill of materials, which is essentially just that list of all the things you need to make whatever product you're trying to put out there. And essentially, based on what we do know, because we do have a good amount of knowledge and development as is, so we're taking that and we're putting together this tool to ask a lot of questions about the materials to kind of challenge them to see if they are where they need to be to proceed to commercialization. So we wanted to make this interactive tool that includes all these different flow diagrams and these decision trees to really get down into those different factors. And we also we suggested timeframes. So some factors are going to take longer than others, and I'll go through that in the next couple slides.
Yes, so just to say this was a publication, the tool that Monica described was a publication that's just been available to download on the BioPhorum website.
Okay, I think I will talk about the pre-screen tool we developed. So I will talk about how this tool may be applied by the user and how the team developed the tool. And then I will share a few examples that we use to stress test this tool and to see if it can produce the same output that we are expecting. So yeah, I can start from the tool itself. So this table is a simplified view of the bomb pre-screen tool. The purpose of this tool is to help user quickly identify materials that has high priority that need to be tackled first. And significantly less amount of time and effort is needed to complete this assessment comparing to a more formal and detailed risk assessment for raw materials. So for this one, each material has 5 questions, and after the user entered the response to these 5 questions, the tool will generate a score, and the score is grouped into low, medium, high priorities. And in order to simplify the tool, we also designed the question to be yes/no type of questions. And from here, we can see that material 3 would be the first one to focus. And then the next step is the user can identify follow-on activities from this assessment. As you can see here, question 1 and question 5 are highlighted. And associated with these questions, we have flowcharts. User can use this to consider the activities as priority. Like each question is associated with a certain flowchart, which we will discuss more in the later slides. And the third information the user can get from the tool is estimated time frame for each of the activities listed in the flowchart. And if additional information is needed, for example, like material attributes, they are listed in other BioPhorum publications. That is also very useful that users can look up as a reference. So now I can discuss how team achieved what we have. So the first step is for us to, in a few working sessions, brainstorm a rather exhaustive list of activities need to be completed to have materials ready for pivotal or commercialization. Then we allocate these activities into different buckets. Eventually, we identified 6 buckets. So these are all 6 buckets identified. So we can start from critical material attributes. So these are the attributes for material from each material that they may have impact on the product quality. And from here, it's associated with assay qualifications, and the assay qualification lead to QC site selection. On the supplier side, we have supplier qualification and the secondary sourcing, and lastly, we have research material risk assessment bucket. And the next step is after we have identified buckets, in each bucket, we have key activities and the decision points. And from these, we can create corresponding flowcharts, like map out the decision trees for each bucket. This is an overview of all 6 decision trees we have created. I think a more detailed discussion is in the white paper, and I think we will talk about one of those in later presentation. The third step is estimate time frame. So for each activity in the flowchart, team came up with an estimate time frame for this activity to be completed. So this is an example using critical material attribute. You can see in this flowchart we have 4 boxes for activities. Is there a supply, material variability, material characterization, and the material impact? And we did this via survey. So the team members will receive survey to get a poll on how long team member think each activity will take, and then we will get together and get alignment on how long the time should be. And then we put the time frame in the box, like here highlighted in orange. Easy to see material impact. Does the material attribute have a direct impact to CQA and CPPs? Would take roughly maybe a year to a year and a half to complete. So this one became a critical activity to complete that takes the longest. And then it became one of the questions in the tool. So we repeat this for the other buckets, and eventually we identified 5 critical activities that take the longest time. We turn these activities into questions in the pre-screen tool. This is this key question highlighted in the flowchart. Okay, so this is a list for all the 5 questions in the BOM pre-screen tool. So we start with, is this a research use only materials? So ideally, we want to use materials manufactured under GMP conditions. Sometimes for ATMP, the material is as new as the therapy itself. So off the shelf, we can only find research use only materials. And in order to convert the RUO material to GMP material, it may take significant amount of time. So this become one of the question. And the second question is material well characterized, understood? What we are asking here is, do we know the impurities? Does it have a limit for purity? Does it have a monograph? Is ID testing available? And the third one is the one we just went through. Does this material possess attribute which could have a direct impact on product CQA or CPPs? And the fourth one is supplier related. Is the supplier qualified and currently active? So different places may have different procedures, but in order to qualify a supplier, you may have to assess previous experience, like supplier performance audit. After audit, there is follow-up actions to complete. So this may also take significant amount of time. And lastly, we have are the material release testing methods qualified? So some of these material, we may need to develop and establish testing methods, which also is time consuming. So these are the 5 questions that we think is critical and take the longest. So we include in our tool. This is a closer look at the tool. So in the tool, we designed 2 columns that user can enter free text. User can enter like material name, or if you already have internal product material code and descriptions. And each question has its own weight, and the score is generated in the end. For each question, user can enter yes, no, or unknown. So in terms of the priority levels, we decided that lower than 5 would be a low priority, 6 to 15 would be medium priority, and 16 to 25 would be high priority. And when there is a box that is not entered, we will remind the user that there is a data missing, show as incomplete calculation here. So here is the fun part. So the team tried a few real-world examples to see if we can get expected outcome. So we tested a growth factor that is research use only off the shelf. It is procured from a new supplier, like small, less established supplier, and is also single source. The second material is a dissociation solution. It is a well-characterized material. There's no known impurity, and supplier is to be qualified. And the third one is a formulation buffer. It is purchased from well-established supplier. It is well-characterized material, and it's manufactured under GMP conditions. So we can have a maybe a second to think about the answer and see if the output matches what we think. So yeah, so this is the output from our tool. So the growth factor is the highest, has the highest priority, given that it's RUO material, it's less established supplier, and it's single sourced. So the dissociation solution has a medium priority, given that the action need to be completed is to qualify the supplier, and the lowest priority is the formulation buffer. So yeah, so basically the team think the tool successfully identified the priority levels for each material, and it met our expectation on the tool.
So we have 6 decision trees, and that is a lot to go through in the time that we have. So we were going to do a quick poll to give y'all options between the 3 decision trees to kind of discuss which one you would like to go through. But yeah, so in order to answer each of these questions, we have each of these is given a flow chart that has some more detailed questions to help you arrive at that yes or no answer. So therefore, if you're not sure like where you're at with it, then you can use the flow charts to help you determine it. So these are the 3 main questions that we chose to let y'all kind of pick between which one you want to talk about more thoroughly. So the options are either the research material risk assessment. So if you're dealing with a research material, it kind of depends on how well known your material is. That kind of determines that risk. You can look at your critical material attributes, which has to do with what aspects of your material impacts your process. Or you can look at assay qualification, where, is your testing GMP and commercial ready in terms of it being applied to quality releasing your materials for use. Okay, so when it comes to critical material attributes, so the first question that we have on here is there supply? So when you're dealing with a material, there's something called the pharmacopoeias, and they're generally region specific. So you have the USP, which is /NF, which is like North America and the US. You have the EP, which is Europe. You have JP, which is Japan. And essentially what those pharmacopoeias do is for any given material or chemical or compound, it has a standard list of testing. So it can be ID testing and impurity testing and a few other things. These pharmacopoeias are very useful because while they're not, at least in the US, while they're not technically like law in the regulations, the FDA expects that we follow them. Again, at least in the US, I think it's pretty similar in Europe and Japan too. But this is really useful to know because a lot of materials, if you're dealing with very simple compounds like hydrochloric acid or sodium phosphate or whatever, you'll probably find that in the USP, and that will basically give you the answers you need in terms of what testing is required, what impurities you need to look for. If your process has certain demands, like there's a very hyper-specific attribute on your product spec that the material can affect, you are expected to look at that as well. But generally speaking, if there is a monograph for it in your pharmacopoeia, then it's like being given the answer on your exam. It's really nice. So ideally, you want a material that is available across multiple grades, which means your supplier is testing that material against multiple monographs or multiple pharmacopoeias. If that is not available, and this is usually the case if your material is like very new and novel, research use only, or if the material is more complex. So if you're dealing with some kind of mixture of like multiple different compounds or whatever, you're not going to have that monograph to give you the answers to your test. In which case you kind of have to figure that out on your own or find a supplier that does have the material available in the grades you need. And that would go into your secondary supplier. And secondary supplier would be like if supplier A has your material available in USP, but supplier B has your material available across like USP and EP, and you're looking to bring your product to multiple markets, you're going to want to go with the second supplier. And then you also want to see if there's suppliers that can offer, or you want at least 2 suppliers ideally that can offer your material in multiple grades. But either way, so that is the best case scenario. If you have a multi-compendial material. Worst case scenario, your material's brand new, novel, no monograph, and you have nothing to go on. Your next thing you want to look at is material variability. So how consistent can your raw material be produced? Can it be produced to the same grade and quality across every single lot, or is there going to be variability? I think with variability, the more like organic it is in origin, so like cells or maybe things that are derived from animals or whatever, the more variable it potentially might be. So for example, if you have like a certain cell line you want to use, or I think or other like peripheral cells, or you could be looking at media or whatever, things that are more organic in nature, you could end up with more variability across your lots, and that's something that you'll have to control for. Whereas if you're dealing with a fully synthetic chemical like sodium phosphate, sodium phosphate, the only thing you have to worry about is making sure you're producing it with as few impurities as possible. So it's a lot easier to maintain like a more homogeneous or consistent lot quality in that case. Your next question is going to be material characterization. So are there certain impurities that the material will have? Are there, how stable is it when it's stored on a shelf for, 3 months, 6 months, 2 years, 10 years, whatever? Are there any toxins that could be in that material that can show up in your product? Are there any residual solvents that are used to produce that product? Just all the, basically you want to know how pure your material is and how stable it is and what effects it could have on your product. Because whatever impurities, or residual solvents or toxins or whatever is in your raw material has the potential to pass along into your product. So those are things that are very important to control for. The next one is material impact. So if you have, a product that is, I think a good example that I've come across recently with the FDA is a DEG and EG, diethylene glycol and ethylene glycol. There's certain compounds that can degrade into those 2 byproducts, and there's certain products where you don't want any of that in there because it can have some, I don't know, it can induce some sort of immune response or whatever it might be. You basically need to like specifically check for those impurities because they can pass on to your product and cause problems. So depending on how well you know all these factors about your material determines how critical it is and whether it's going to add time to your timeline. So if you have a research use only material that's not well characterized, you don't know anything about the stability, it's going to be a lot harder because you're going to have to do a lot of risk assessments. You're going to have to do additional testing. And it's going to take a lot more effort to qualify it. Whereas if you have a USP, EP, JP grade synthetic chemical that you know how this shelf life of it and you know its impurities and you know all these things, well, then you're good to go. So it just depends on how well you know your material and how much it'll impact your product.
Okay, I would like to cover the phase appropriate requirements. And as everybody knows, as you go into the clinical phases, you have 1, 2, and 3, all the way to commercial. So we want to discuss that next. Here shows the different requirements in each phase. And as you'll notice in phase 1, you're just required to do identification testing on your raw materials. And at that point, you want to make sure you have a BSC TSC. If you notice at the note below, that's for protection of your site so that you don't get it contaminated, possibly. And as you go into phase 2, you need to have some additional requirements. One is a supplier quality system in place, your audits, your quality agreements, to make sure you have a locked-in supplier. And also, you need a safety data sheet and supplier statements for anything that might impact your process. And that you also want to know if your process and your raw materials are stable. So you will do some kind of stability to make sure nothing's going to break down. And the next phase, then you're going to phase 3. And at this point, it's the same as phase 2 as far as material testing. So it's still just identity of your raw materials at that point. But at this point, you want all your suppliers qualified, fully locked in, so you know you can depend on that supplier, submitting to your final product. That is pretty much a site audit for all your critical materials. And as you can see, the documentation and stability are same as phase 2. And on your potency or anything that's required for your process, that's applicable to your specific process. So if you need some kind of potency verification, you would do that on your own based on your process. And as Malika mentioned, you want more than one source once you get close to commercial. So in case you have an interruption, you don't mess up your product life cycle. So you do want materials dual sourced at that point. And your next step you go to is when you get close to commercial, we call it pivotal. And that sometimes could back up to phase 3 for some companies, but it's not required at phase 3. But at the pivotal commercial step, before you go there, you want to have full C of A testing. So at that point, it's not just identity. You'd have to test all your impurities and your assay results to ensure that your raw material maker is under control and trending within specifications. You need evidence that they're adherent to GMP. You need some kind of a GMP certificate. And on your supplier documentation, you need the C of A test methods, because if it's not compendial, which is your first option, you want a compendial material so you can use the USP, EP, and JP methods. But if you don't have a compendial material available, sometimes you have to go with a custom material, and that's when you would get the vendor methods that they use to test their C of A, and you would have to, at receipt, verify their testing results. And on stability data, that's the same as phase 2. And on your potency and all your dual sourcing, definitely that would be required also. Now on this slide, you see all the way to the left is your phase 1 and 2, and that's what you would want to confirmatory test on your supplier's raw materials. And you need, of course, identification. You need their C of A, but also you'd want a BSC TSC statement, because that's not as much about the raw materials, it's about protecting your processes, your equipment, and your site. So you make sure it don't get contaminated. So you want that early on up front, because without that, that material might not be able to go to full commercial use anyway. So then as you go to phase 3, it's the middle example. You still do identification testing, like it says, and appearance, because at that point you want to start looking at your material, make sure there's not any particulates, and make sure no color changes. So you're starting to look at your material closer here. And you'd want to, of course, your BSC TSC, but at this point, you'd also want to know critical impurities. So if you have a raw material that might bring some critical impurities into your system, you would need to know that. And if you don't risk assess it away, you'd have to have some drug product testing to ensure that you removed it if it was in that raw material. And then you just need to know more of a GMP that you would go to commercial with it. And as your last example here is your pivotal commercial phase. You have, again, just like phase 2, you'd have your appearance identification testing. You'd have BSAT, of course, but at this point, you also go to purity testing to make sure it is pure, that meets the specifications. All your impurities for the raw material at this point would be considered, not just the critical ones that you would need to show drug substance removal. And the last part of this is a very odd requirements. You have to have melamine statements and nitrosamine statements. At this point, you'd want to protect the patient from any consequence of these raw materials. And melamine is one where if you have in your raw materials a requirement to have a high level of nitrogen, like if you need a high level of protein or nitrogen, you would need to do a risk assessment because there's an economic incentive to spike it with melamine. So that's a concern you need to be aware of. And also a GMO statement to make sure it's not genetically modified. And another one would be, of course, residual solvents. If you have any raw materials that say they use a solvent, you would have to test for that solvent to ensure that it's compliant within the, and that's based on the supplier's statements. So these are the different levels we recommend that you would, as you go in your product life cycle, the commercial, that you would test and to be concerned with. The conclusion is experts from 12 organizations collaborated to assist how to best prioritize your raw materials. This team created a tool that we've gone through earlier, and is to screen all of your bill of materials and associated concerns so you can determine the timeline and what testing to prioritize. And this high-level screen can be used to quickly review, just triage every material in your product. We also included, as I just discussed, the phase-appropriate recommendations. And this tool also be used by any professional working in raw materials or at a site where raw materials are entering your process to ensure you have the right raw material and then you're taking care of all the concerns for that raw material.
So the question is, how do you know what is required for testing based on each development phase? I don't know if anyone wants to take that, Dave.
Since I just covered that, I think it's very important for you to know where your product life cycle is at, because early on, when you're developing your process, you don't know if you'll use that exact vendor and material. So you just do an identification test to make sure it is what it is and you're using the proper material, but you don't have to have all the trend and attributes and every impurity at that point. And you only need that when you say this is a commercial product, or my product is locked in. And at this point, you would have to do all the testing. And that's why they developed the phase 1, 2, and 3 before you go to commercial.
Thank you so much. I'll let you go. Thank you, everybody. Appreciate that. And talk soon.